When it comes to radioactive substances in the environment, there's a fascinating interface with biology, the interaction of radionuclides with microorganisms. As in the last video, the topic is final repository for high-level radioactive waste. In the last video, we looked at the interaction of radionuclides with minerals, and today we will look at microorganisms. So what's on the agenda? What is high-level radioactive waste? Why do bacteria play a role in this rocky environment? And in the practical part, we will look at how to set up such an experiment and then talk about the results that could be drawn from experiments like these. High-level radioactive waste is produced in large quantities in the energy sector, but there are also research reactors and, in a broader sense, reprocessing plants, laboratories and so on. In the simplest case, we are talking about spent fuel rods. During the reactor operation, the uranium-235 enriched to approximately 3.3% is fissioned, and new elements are bred from the remaining 96.7% uranium-238 partially through neutron capture. After usage in a standard nuclear power plant, we are left with only 0.9% of the 3% uranium-235, which corresponds to a burnup of approximately 70%. The fuel then contains 94.5% uranium-238, 0.5% plutonium and 0.1% actinides, including the bred transuranic elements, i.e. the elements that come after uranium in the periodic table, so 93, 94, 95 and so on. Plutonium element number 94 is also included but has been given its own category, and the fuel now contains 3.2% fission products. Many of the fission products can be weighted out and then, as in Finland for example, packed in canisters and transported to a deep geological repository with a multi-barrier system. The multi-barrier system has the task of isolating this mixture of radionuclides from the environment for at least 200,000 years. 200,000 years is the time that it takes for the activity of the high-level radioactive waste to resemble that of naturally occurring uranium. In the course of this, roughly 10 half-lives of the plutonium-239 have passed, and 10 half-lives is in practical radiation protection the time after which we say that the radionuclide is completely decayed for all practical purposes. Germany's requirements for a final repository are that it must be able to isolate the radionuclides from the environment for 1 million years. As part of this multi-barrier system, many different systems are being investigated in the case the worst case scenario occurs, namely that the radionuclides escape from the canister. To do this, the radionuclides must first pass through the uranium dioxide fuel itself, then through the zirconium dioxide cladding, and then through the cast iron filling, and finally through the copper wall of the canister used in Nordic countries. Once this has been achieved, the radionuclides are now considered released but still isolated. They will now encounter the bentonite clay. Microorganisms can live in this clay. These microorganisms are anaerobic in nature. In the case of Finland, they must live at a depth of 500 meters below the Earth's surface. So they need to produce energy without the aid of oxygen. For example, we humans mainly use aerobic processes to provide energy. Aerobic means that we use oxygen. We obtain oxygen by simply breathing it in. It makes up approximately 20% of the air. The oxygen molecule can then precipitate in metabolic processes as redox active compound, and then ultimately the oxygen atoms of the inhaled oxygen are present in the form of water with a change in the oxidation state from 0 to -2. Energy production is extremely complicated, but roughly speaking, we need a redox active compound for energy supply. We can see from the simplified reaction equation that oxygen acts as an electron acceptor or an oxidizing agent. And we usually use forms of sugar as an electron donor to obtain ATP via redox reactions and so on. Now there is no oxygen available, so other electron acceptors are needed. Possible candidates are nitrate, which becomes nitrite, manganese 4+ to manganese 2+, iron 3 to iron 2, fumarate to succinate, as well as sulfate to sulfide, and several others. The complex chemistry of microorganisms also produces other compounds known as metabolites. And it is not only these metabolites but also the membrane of the microorganisms themselves that can potentially interact with the radionuclides that have made their way into the bentonite clay in our context. There are some compounds that make metal ions more mobile through complexation. Most radionuclides, or elements in general, are metals. The now soluble complexes with the radionuclide metal ions can spread more easily than the metal ion in its original solid form could. That is not what we want today. We want these microorganisms to immobilize these radionuclides so that they do not spread any further than they already have. They can absorb the radionuclides on the surface of the cell through functional groups such as amine, hydrogen phosphate, carboxylic acid, or hydroxide groups. This is called bioabsorption. Or they can also absorb these elements. This is called bioaccumulation. Other metabolites such as sulfides, phosphates, and or carbonates can form poorly water-soluble compounds with external radionuclides. Metals, sulfides, carbonates, and phosphates are usually poorly water-soluble, which is good for us because solid compounds that do not dissolve do not travel far. Another factor that is highly important for the immobilization of radionuclides, especially if you take a look at the scientific publication accompanying today's video, is bioreduction. Some oxidation states are more mobile than others of the same element. There are various reasons for this. For example, because they are differently reactive. And all these complex forms of interactions between microorganisms and radionuclides are of great interest because they in particular help to isolate the radionuclides of highly radioactive waste from the biosphere, i.e. the environment. But how do you conduct such experiments with radioactive elements and bacteria that also must not be exposed to atmospheric oxygen? There are several research groups working on this at the Helmholtz Center Dresden-Rossendorf. We are currently in the incubation room where the microbes are grown. They do this at around 30 degrees C in a serum bottle like this. Here we can see a culture of Desulfitobacterium. This belongs to a class of organisms which, as the name suggests, use iron as radioactive compound to produce energy. It can also probably use sulfide as an additional electron acceptor. The bacterium was isolated from bentonite and now lives in a medium to which vitamins, pyruvate, sodium thiosulfate as an electron acceptor and trace elements have been artificially added for optimal growth. This was added after autoclaving the vessel, as some elements could precipitate during this autoclaving and thus no longer be bioavailable, so not available for the bacterial growth, which we want. However, the dark stuff here was a cell suspension, not precipitated elements. This was just a little digression on how to care for bacteria. You don't really want to have such a biomass for your experiments. With a curve like this, which actually describes the bacterial growth, you can also indirectly map the bacterial activity. You can see that at some point in the stationary phase, there are so many bacterials that there is no more space or other resources available. You don't want that for your experiments. You want really nice active cultures like here. This is another anaerobic bacteria species, namely Pseudodesulfos vibrio aspergensis. As an anaerobic bacteria species, it uses the redox system sulfate to produce hydrogen sulfide. This culture was freshly prepared and now we want to take a look at it under the microscope to see if they are really active enough for further experiments. The problem with oxygen can be easily circumvented with inert gases such as nitrogen or CO2. First, everything must be disinfected with ethanol. The syringe is first rinsed with inert gas. Now, after a brief flame sterilization, a sample can be taken. This sample is only used as a representative of the culture in the bottle. From now on, they are unusable for our experiments. To be on the safe side, we can also take a look at a second culture. Once on a sterilized microscope slide, one culture was indeed more active than the other. So after checking that the microorganisms are active enough and washing them from their growth medium, we can conduct the first experiment. Such experiments are called resting cell experiments. Survival is still a thing and lactate was added, but we only want to look at the interaction of living microbes with the radionuclides. Since we want to take a look at a paper on the immobilization of uranium, we have recreated a urinal solution with a vitamin mixture, which is only added to the microbes with an inert gassed flushed syringe. Now the experiment is done. Well, you usually have to take aliquots, i.e. samples, after 2, 4 and 6 hours on the same day and then leave the microorganisms to incubate for another 48 hours to 1 week. While the microbes are incubated, we can take a quick look at how such solutions are degassed. This is because the urinal solution, the autoclaved growth medium and everything else must be completely free of dissolved oxygen. You simply remove it by bubbling nitrogen gas through the whole thing at bit overpressure. A cannula is used to equalize the pressure, and after 20 minutes not only has the oxygen be removed, but there is also a cushion of nitrogen overpressure above this solution. Now let's take another look at the microbes. They have now had the time to interact with the radionuclides and do some chemistry, and hopefully we have less free radionuclides in the solution and a lot of radionuclides in the microbe pellet. To do this, the bottle and the 2 flasks are then placed in the glove box. After centrifuging, the result is a pellet like this. The liquid, which we hope is less contaminated with uranium, is poured back out for now, but the uranium content is still measured via ICP-MS, better known as inductively coupled plasma mass spectrometry. This allows us to detect very small trace elements with a high degree of reliability. The pellet with the microbes and hopefully a lot of radionuclides is then dissolved in 1 to 2 milliliters of 5 molar hydrochloric acid. And now at the latest, the microbes are dead. So they have survived for a while, but in this case the microbes have 2 possible causes of death. Either because the uranium was added, however the cause of death is not the radiation, but as I keep saying, the danger of uranium is primarily not the radiation but the fact that it's a toxic heavy metal, which is what might have killed the microorganisms, or at the latest the hydrochloric acid washing step. After waiting 2 to 3 hours, all of the uranium is dissolved again. The supernatant is then measured via UV vis. Optionally, a pellet not dissolved in hydrochloric acid can be measured using TRLFS, which we will look at tomorrow. Here's a quick digression. TRLFS stands for time-resolved laser-induced fluorescence spectroscopy. The measurement method is used for samples that can fluoresce, and uranium can fluoresce under certain conditions. This you might know from uranium glass, but a lot of important information can be gained from this fluorescence. Elements other than uranium and other compounds can also fluoresce, but today we are primarily concerned with uranium. If you would like to know about this, there's a video about TRLFS, and this video will be linked here. The next day we must continue with the same step. The supernatant is drawn for the ICP-MS. These 2 ICP-MS samples must be mixed with a volume percentage of the sample in concentrated nitric acid for measurement. Another aliquot for the liquid TRLFS is also taken. With the TRLFS we can not only determine how much uranium is present in the solution or the microorganisms, but also make statements about the chemical form based on factors such as lifetime of the uranium fluorescence and also band shifts. Since we also want to know how it is contained in the microbes, a pellet is also needed for the measurement. The suspensions were combined and centrifuged. In the meantime, we can take a look at the sample holder. It is basically copper with a quartz glass disc and a frame. And now that we have a pellet, it can be pipetted into the frame and carefully screwed on. The quartz disc costs a mere 80 euros per piece, and for TRLFS measurements, it goes in a sample holder like this. And then hopefully we will get new insights, but what kind of? This video was inspired by a recent scientific publication by Stefan Hiltmann et al. In the underlying research, the Desulfitobacterium species G12 culture shown at the beginning was used to immobilize uranium. Desulfitobacterium belongs to the iron-reducing anaerobic bacteria. This means it uses the iron 3+, iron 2+ system. To give a little background on this paper, in a previous paper it was shown that another bacterial species called Desulfosporosinus hippii DSM 8344T can reduce uranium 6+. But only if the hexavalent uranium is present as a uranyl ion in the form of a lactate complex. This lactate complex really does occur in pore water of certain clay minerals. And if that is the case, then Desulfosporosinus hippii reduces this uranium and immobilizes it due to the lower mobility of uranium 4+. The extremely cool thing about this is that part of this reaction involves pentavalent uranium being present as uranyl 5 monocation. This oxidation state of uranium seems to be more relevant than I personally assume before making this video. Why am I saying this? Well, as we have already learned in this video, carbonate complexes are also of great importance when it comes to radionuclides in the environment. And this sulfate-reducing bacteria species was not able to reduce the famous tris-carbonate uranyl 6 complex. This means that they went ahead and looked for another bacterial species, preferably one that might not use the sulfate-sulfite system, and now we finally come to the Desulfitobacterium species G12. Because there you can see quite clearly that the uranium was immobilized, not just in any way, but the form of the tetravalent uranium. Here's our supernatant, which should contain little uranium. In the case of Desulfito, 60% of the originally dissolved uranium was no longer present after 48 hours. And after a week, as much as 90% was no longer present. Dead bacteria only showed a 10% reduction in the uranium concentration from the supernatant. This means that uranium immobilization is really an active process that can only take place with living bacteria and is not a passive process of sticking to the membrane of a dead microorganism. And in this case, it only worked with uranium present as a carbonate complex. With a lactate complex, it was less successful. The UV-Vis also showed with green and orange as reference for the respective uranium oxidation state that the immobilized uranium from the carbonate complex is indeed present as uranium 4+. And these transmission electron microscope images are also very nice because they show that the immobilized uranium is really present inside of the cell. Further analysis were carried out, but we have already discussed the core content. The Desulfitobacterium of the species G12 is an anaerobic bacterium that uses the iron 3+ to iron 2+ system. In addition to providing energy, this system can also allow the bacterium to immobilize dissolved uranol in the form of uranium 4+. Which is found inside of the cell after this redox reaction. It was also shown that after a week of incubation, 90% of the dissolved uranium could be immobilized that way. However, this is only possible if the uranium was present in the carbonate buffer solution as a tris carbonate complex. When present as lactate, this type of bacteria is less effective in terms of immobilization, but there is also another type of bacteria that can do so, the Desulfosporinus hippii. That is really cool. Not only can different types of bacteria selectively reduce different uranium species, but also bacteria can play a role in the context of final repository. And there are so many possible types of bacteria and other chemical forms of different radionuclides. So there's still a lot of research ahead of us. A big thank you goes out to Dr. Stefan Hildmann, whose paper I just mentioned, and to Dr. Marcos Felipe Martinez Moreno, because these two did all of the practical work for this video. And it all took place under the supervision of Dr. Andrea Czerkuk, head of Deep Biosphere Research at the Institute for Research Ecology at the Helmholtz Center Dresden-Rossendorf. And a big thank you goes out to my patrons for the financial support. With that being said, thank you for your attention and goodbye.